Good afternoon. I'm Judith Greenberg, Acting Director of the National Institute of General Medical Sciences, and I would like to welcome you to our annual DeWitt Stetton Jr. Lecture. Our speaker today, Dr. Russ Altman of Stanford University, has broken new ground in applying computing technology to medicine, primarily in the area of pharmacogenomics. He began focusing on the intersection of computation and medicine long before it was trendy, during his graduate studies at Stanford. This positioned him well to mine the profusion of data that has emerged from the Human Genome Project and a wealth of other basic science and clinical sources. Dr. Altman's forte is integrating different layers and types of data into a coherent picture that suggests hypotheses and experiments to test them. He also develops and applies bioinformatics tools in novel ways that reveal conceptual and physical relationships. Dr. Altman received an AB in biochemistry and molecular biology from Harvard College, a PhD in medical information sciences from Stanford, and an MD also from Stanford. He became a Stanford faculty member in 1992. In addition to his own research, he directs two NIH-funded projects: the National Center for Biomedical Computation at Stanford, which focuses on physics-based simulation of biological structures, and the Pharmacogenomics Knowledge Base. Dr. Altman's honors include a Presidential Early Career Award for Scientists and Engineers and election to the Institute of Medicine of the National Academies. I refer you to the program for further details about his impressive background. I'm proud to say that NIGMS has supported Dr. Altman's work for more than a decade. Before I invite him to the podium, I'd like to take a moment to tell you about the highly regarded scientist for whom this lecture is named. Dr. Stetton spent most of his career at NIH, serving in two institutes and in the office of the NIH director. He was perhaps best known as NIH Deputy Director for Science, but prior to that time, he served as the third director of NIGMS. In recognition of his many contributions to NIH and to the scientific community as a whole, Dr. Stetton had the rare distinction of having two NIH entities named after him. This lecture is one of them, and the other is the DeWitt Stetton Jr. Museum of Medical Research, which was dedicated during NIH's centennial in 1987. Dr. Stetton died in 1990. And now on to Dr. Altman's talk, which is entitled The Emerging Network of Data for Understanding the Interactions of Genes and Drugs. Dr. Altman.
Thank you very much for that nice introduction, and I've had a great day visiting particularly NIGMS, and it's very good, and thank you for coming even in the rain to the talk, and hello to the TV land out there. I understand there's thousands of you, hundreds, tens, whatever. I just want to say one word about Dr. Stetton. Of course, you know, when you are invited for a distinguished named lecture, if you're smart, you find out about the background of the person. And there's many things that you just heard about. The few things that I want to highlight that really struck me and sounded a little bit familiar is that he was born in New York in 1909. He did his MD at Columbia University, and he did his internship and residency at Bellevue Hospital, which is exactly true of my grandfather. So I think there is a chance that my grandfather and Dr. Stetton knew each other. I don't know that for a fact, but those three facts are definitely true for both of them, and so it's possible. So that was funny because my grandfather just would have had, he passed away a few years ago, but he would have had his 102nd birthday yesterday, so I thought it was a nice little connection. So I'm very honored. I do want to thank, I'm going to do my thanks up front. I want to thank NIGMS. They have been supporting my work for more than a decade, but if you include the fact that they supported my MD and PhD training through the MSTP program, then they've been supporting my work for 3 decades, for which I'm very grateful. And then you've heard about PharmGKB Symbios. We have a computational training grant, have had one at Stanford. I also like to thank National Library of Medicine, and most recently NINDS has made, for the first time, I'm part of an award from them. I would like to acknowledge gifts and also some conflicts which are really, I don't think, relevant to today's talk, but I list them anyway. And I will put my phone on mute. Okay. And I want to thank my coworkers. This is some of the people in my laboratory who work very hard. Almost all of them have a project that involves drugs. That's why I'm talking about drugs today. And you will be hearing about the work of the people I've circled and whose last names I've provided on this slide. And of course, everybody is working hard, and I'm grateful for all of their efforts. Okay. So I want to outline today what I'm going to do. In our laboratory, we have a focus on drug action very broadly at the molecular, cellular, and organismal level, with applications particularly in pharmacogenetics, understanding drug mechanisms, repurposing, using old drugs for new purposes, and discovering drug interactions. And our toolkit, and this is, I think, what makes us a little bit different, is entirely computational. I have space, but I do not have a bench. So if there are experiments to be done, it's because I take an experimentalist out for coffee and convince them to do them, or I pay somebody to do them. But they don't happen in my lab normally. So we are really relying on using data and computational techniques to generate hypotheses, but also to at least develop initial proof that these hypotheses are reasonable. And I'm going to tell you about, and we do this for discovery and for leveraging the investment. What I mean by leveraging the investment is there's a sense in informatics that many valuable data sets are not completely mined for the nuggets of gold that they have within them. And I want to tell 4 stories today of data that was collected through high throughput data collection experimental efforts, published by others, and then we've kind of gone back over and found some interesting things. And I think that really the message that I'd like to get out today is these informatics technologies, some of which are simple, some of which are complex, we'll have a range in the talk today, but they're all potentially extremely powerful for understanding what's going on. And they're even more powerful in concert with one another. And the title of my talk was something about a network of data. That's what I mean. Any given modality may give you a little bit of confidence that something is true, but when multiple modalities start telling you that something is true from an independent perspective, then you really begin perhaps to believe it. So the 4 stories I'll tell today go from the molecular to the population base. One of the beautiful things about not having a bench and not having any equipment is we can look very broadly at data from multiple sources without having to retool the lab. So the first story is about structural molecular data and drug repurposing. The second story is going to be about the use of expression data to understand cellular mechanisms of action. Then I want to talk about text mining as a way to predict drug interactions. And then I'm also going to discuss drug interactions in the context of mining population-based databases and electronic medical records. So really we're going to go from the molecular to the population. A lot of this interest comes from the 2 projects that are NIGMS funded that I want to just briefly mention. The first one is the PharmGKB, which is the pharmacogenetics knowledge base. We have the goal of categorizing and collecting all information about how human genetic variation impacts drug response. We've been in business for about 11 years. This is a website. I'm very proud of it, but I'm not going to talk about it. But this is kind of the one-stop shop for the effects of genes on drug response, and I encourage you to visit it at pharmgkb.org. Because one of the things I report every year is the number of hits on the site. So please hit it from home, from work, from your PDA, as much as possible. But the key point about pharmacogenomics is that in many ways I think this is a very important goal for medicine, because you can only do pharmacogenetics really when you understand the details of drug action. So this is a pathway for how warfarin works that you can find at PharmGKB. This is the pharmacodynamics pathway. We also have a pharmacokinetics pathway, and if only we had these kinds of pathways for all drugs, then we would know all the molecular players that modulate the response to a drug. With that information, then, we could then look for variations in the human genome that change this response, and therefore we could do very robust, personalized prescriptions based on genome and other information. So, for example, on PharmGKB, now you can go and you can find out that there are 2 genes for warfarin, CYP2C9, a cytochrome P450, and VKORC1, which is the target, and that there's SNPs, single nucleotide changes in DNA, that have specific impacts on warfarin dose. In particular, if you have this particular SNP in the cytochrome, AA has an average chance of response to warfarin, where CC is associated with a decreased dose of warfarin. So the vision for pharmacogenetics, and really for all of understanding of drug action, is to understand the molecular mechanism and even how variations modulate that mechanism. And so much of what we do is driven by this long-term goal. We recently used PharmGKB to do a complete annotation of a single human genome, and really the headline there for you is that there are hundreds of drugs that we felt like we could come up with clinical advice. This was published in Lancet last year, and I'm not going to go through the details, but I'll just say that this is not a 1 or 2 drug field. This is a field that has evidence for hundreds of drugs that we were able to collect in annotating a single human genome. We recently published another analysis of a family quartet, a mom, dad, daughter, and son. And again, I'm not going to talk about it, but I'll just show this table from the paper, which are variants associated with adverse drug response. And we have dad in dark blue, mom in pink, in dark pink, boy in light blue, girl in pink. And really this just shows that genetics is as advertised. Sometimes the whole family agrees because they all have the same genetics, but sometimes mom and dad and son agree, and mom and daughter are so more likely to cause a side effect versus less likely to cause a side effect. And we can do this for many, many drugs. So we are making good progress in pharmacogenomics, and there's still work to be done. All the way at the other end is my interest in 3-dimensional structure and function, and this is what Symbios does. It's a National Center for Physics-Based Simulation, and one of the things we study there is G-protein coupled receptors. G-protein coupled receptors, this is the beta-2 adrenergic receptor. These are targets, as many of you know, for a large percentage of drugs on the market, but in many cases we don't have a deep understanding of how the drug binds or how it affects the conformation to create the signaling pathways. We'd like to understand that. And so one of the things we do at Symbios is put a GPCR, which you can hardly see here, in a lipid membrane with water on the internal and external parts of the cell, and simulate the dynamics in order to understand what the motions are that may help us understand the structure and function. Okay, so now I want to tell you these 4 stories. Let me begin a little bit with an apology. By its nature, 4 stories will be short and will not go into a ton of technical detail. Almost all of this work is published, but in order to get the big picture, I needed to go up to a little bit higher altitude. And so I apologize for anybody who finds this technically unsatisfying. So, Structural Genomics Initiative has been a big push in the last 10 years, led by NIGMS, and the goal was to get an increase in solved 3-dimensional structures, and this has been fairly successful with about 70,000 3-dimensional structures in the protein data bank now. And many of those structures are complexes of proteins with small molecule ligands, including drugs. And so a question you can ask is, can we identify or use this information to predict new interactions between proteins and small molecule drugs based on the examples that we have? And I'd like to tell you that I think the answer is yes. So for the last few years, my lab has been developing the feature system for describing microenvironments within proteins. So here you see a protein. This is just a piece of a protein, and I've drawn a blue circle around a microenvironment that's approximately 7 angstroms in radius. What we do is we go to these microenvironments and we build statistical models of them based on the occurrence of various biophysical and biochemical features, such as the types of atoms that are found, these are listed at the bottom here, the types of residues that are found, the secondary structure, the charges, the hydrophobicity, and really a long list of biochemical and biophysical properties, which give us kind of a signature, a computational or statistical signature of that site. We do many things with these signatures. One thing we do is look for other sites that are similar. And we have shown previously that we can detect remote similarities in different proteins that are remote but have similar microenvironments that may therefore have similar function. Now, one of the applications of this for drug or small molecule binding is shown here. So what I have here are 2 protein structures, both of which bind flavins. And you can kind of see one flavin here and one flavin there. I'll talk about those in a second. And then you see what we call the feature balloons. What these are feature microenvironments shown in different colors. And I've chosen the colors on the left and the right for environments that were very similar, based on a very similar pattern of physical and chemical properties. And so what you can see is that there's a blue balloon here and a blue balloon there. There's a purple one, purple, etc. And what we found, and this is not surprising, was that different types of environments were associated with binding different parts of the small molecule flavin. So for example, here you have the adenine, well, let's take an easy one. We have some negatively charged oxygen-rich areas in the middle, which are near the purple and the red, and they're near the purple and the red here. I want to point out that the exact configuration of these balloons is not the same in the 2 structures, but they're always binding similar portions. So you can see the blue balloon is near this 3-membered ring, this triple ring, and that is true here as well. So what we found is that feature was able to detect the similarity in these 2 binding proteins. These proteins are quite distant evolutionarily, but we were able to see the similarities by the common configuration of these microenvironments. This is a particularly hard example because flavins are known to have 2 conformations, the butterfly and the extended. The butterfly is bent. You can tell me if you think that looks like a butterfly. And this is the extended conformation. Those lead to very different configurations of the colored balls, and yet there's a conservation of the colored balls overall, just with different geometric locations. So we wondered if we could use feature now for repurposing to, let's say we didn't have a flavin bound to this pocket, could we recognize that the flavin has certain colored environments that are present here, and therefore there would be a possibility for the flavin to bind? That's the kind of idea for a structure-based repurposing. This also works for GPCRs. We have the beta 1, the beta 2, and the adenosine receptor, somewhat divergent with different functions, but they all have a common core of feature environments that we think are related to the function. The function of these molecules is different. They bind different drugs, but then they go on to have very similar conformational changes, and we think we can identify the environments that are conserved. And finally, and really more directly towards this possibility of repurposing, we were recently looking at kinase structures. These are 2 kinases that are quite diverse in terms of their 3-dimensional structure, and yet we saw a very similar constellation of microenvironments, which led us to believe that inhibitors binding this kinase might also bind this kinase. And now we're going out, I wish I had the results for you, but we're actually working with a company that does kinase assays to test the binding of a known ligand for this protein and see if it binds this protein. And We don't know the answer yet, but we have some confidence based on the certainty scores and our calibration that this is a pretty good guess. So what's the point? The point is, using publicly available three-dimensional structures and some algorithms, we're able to come up with hypotheses about small molecule binding that can get very far before we spend any time or resources doing actual experimental tests. And so this is part of our emerging network. So I want to move now to molecular and cellular data, and in particular expression data. Since about 1998, there's been an explosion of expression data from different cell types and under different conditions, where the level of expression of a gene is measured in a population of cells. And in fact, there are many measurements, and these are kept, by the way, at the Gene Expression Omnibus, or GEO, which is maintained by the NCBI down the street. There are literally more than 20,000 human samples in that database. And there actually has been published already some examples of drug repurposing based on finding common gene expression patterns. In fact, in one case, they took the gene expression pattern for a disease and they looked at which genes were up and which genes were down, and they found a drug that did the opposite. A very simplistic idea that the drug should undo the disease. But then in some in vivo tests, they showed some efficacy and that it was not an unreasonable thing to do. This was in Science Translational Medicine a couple of weeks ago from my colleague Atul Butte. So we wonder, can we understand drug action using this publicly available data, or part of the network, if you will? So you've all seen, I think, gene expression chips, SNP expression chips. This is one sample one. I made it up. But if you look into GEO, you will find, as I said, 20,000 arrays covering all sorts of human biology: normal biology, cancers, rare diseases, diabetes, cholesterol, pick your favorite disease. There's probably an array in GEO that's measuring the gene expression. And we did an algorithm called independent component analysis, or ICA. And a component is basically a hidden signal in the data, and I'm going to take you through a slide to show you the idea behind ICA. This was published last year. So here's a number of arrays, and if you look carefully, you can see some patterns that these arrays are all combinations of these three basic patterns: an apple, an RNA, and then something that looks like a real microarray data set. And what we've done in creating these is we've just weighted them different amounts to create different array results. And the hypothesis we had is that there really are fundamental modules or components of human expression such that all 20,000 of those human experiments in GEO would be some sort of combination of basic biological modules, pathways, call them what you will. So an independent component analysis is specifically designed to pull out such hidden components. So we applied our ICA to the full set. We downloaded all of those arrays, 22,000 arrays, applied the independent component analysis, and defined 450 fundamental components that were sufficient to explain almost all the arrays in the data set. So every array was a combination of these 450 components. And we plot these components here in a two-dimensional plot, and I just do this to show you that when we look at the genes in each of these modules, they are not random, but instead they correspond to what you would expect to see in terms of functional modules. So in the upper left, we have cell division, mitosis, and DNA replication type genes. Over here, we have cell differentiation, epidermal differentiation. We have immune response, cell adhesion. And so this is remarkable to us because we did not build in any knowledge of biology. We simply asked the mathematical question, what's the key set of basis vectors that allow us to concisely express experiments? And when we got those vectors and looked at the highly ranked genes, we found a lot of known biology. I should point out, I know that it's bad to have a cell phone anywhere near a mic. I should also point out that there were some very nice components that had no labels. we didn't have any theory for why all those genes were together. Now, they could be wrong, but it also could be entirely unexplored parts of biology that the data is telling us it sees, but that we don't recognize because we haven't learned that part of biology yet. So as a side note, all of the ones that don't have good annotations, which would be cluster number 201 and cluster number 251, they look just as good as the other clusters that do correspond to biology. And so the hypothesis would be we need to do work on those genes and what their functions are. Okay, so we have these components. What do we do with them? Of course, it's about drugs. We looked at the response to a drug, parthenolide. It's a drug used in cancer. We had CD34 cells from 12 AML patients. This is all in GEO, I should say. This is all in that same data set. And they had samples of these CD34 cells before and after treatment. So the first question we asked was, what components as a group show up or down regulation in the treated and the untreated patients? So this is the kind of picture I'm sure you've all seen many times with treated and untreated. Patients are the columns, but you're probably used to seeing genes along the rows. These are not genes, these are entire components. So for example, component 373 is up, red is up, in the untreated and down in the treated patients, almost without exception. So it's a module-based analysis instead of a gene-based analysis. And here I just have some statistics. And there are about 10 or so of these components that are really markedly different in the treated versus the untreated. But because 373 was so impressive, so red here and so green there, although not entirely, we took a closer look at that. It was the component with the biggest difference, and we could look at the highly weighted genes in component 373, and we could look back in GEO at the diseases that have a high contribution from component 373 when we do that combination of components to recreate their expression. And in a very satisfying result, we get a whole series of ALL and AML leukemias, which are indeed what parthenolide treats. But you wouldn't have had to told us that, because when we analyzed the gene expression changes of parthenolide, they were in the exact same component that seems the most dysregulated in these cancers. So that was a confidence builder. We next could go and build a pathway, and what I show here is a pathway where the red are upregulated and the green are downregulated by parthenolide. And there are some very famous genes here, tissue necrosis factor, NF-kappa B, and a number of kinases. So what does this mean? So now we've moved in an entirely data-based method with really no understanding of, I had never heard of parthenolide before this study, and I'm not an expert on ALL, but by doing this analysis I got my components, I looked at the components, and now I have very specific hypothesis about the pathways that parthenolide is affecting. Any other disease that involves this pathway, especially if that disease upregulates this pathway, would be a potential target for parthenolide treatment. So this is how I can imagine a routine use of expression data to find repurposing opportunities. And the recipe would be take a look at these genes, see if they're in general upregulated, red, and if they are, then consider parthenolide as a potential treatment. So again, that's a short story. We published more about it, but I wanted to give you a flavor for how the expression data can lead directly to repurposing and understanding of drug mechanism in more detail. I now want to move to textual information. So I've talked a little bit about the network of molecular information, 3D structure, chemicals, the cellular expression data. Now let's go look at text. Pubmed holds 20 million abstracts. Arguably, it is where biomedical knowledge is stored. If you had to point to one spot, you would point to the server room on Lister Hill and say that is holding basically the entirety of our medical literature, especially if you had full text through PubMed Central, etc. But as a computational person, I have mixed feelings about text because I like to read it as a human, but it's a disaster for computers. Computers are not good at reading human-written text, and yet that's where all the gold is. So we have to ask this question. I ask it begrudgingly, but I ask it nonetheless. Can we train computers to read the literature and reason over them? And in particular, can we do it to predict drug-drug interactions? And I want to show you that I think the answer is yes. So in the computational science field of natural language processing, or parsing, NLP, there's been some revolutionary advances in the last few years. And one of them is the ability to do very accurate parsing of sentences so that the nouns and the verbs, the prepositions, all the things you might have learned in 5th grade can now be identified by computers with pretty good fidelity. So here's a sentence. The ABCB1 C3435T polymorphism influences methotrexate sensitivity in rheumatoid arthritis patients. That's an actual sentence from Pubmed, which we can parse. You can see it recognizes some things as nouns. It recognizes influences as a verb, etc. And it builds computational representations of the sentence. I will not go through this, although it's a fascinating area where linguistics and computer science, and in this case biology, all meet. But I'll just leave it at the fact that we now are able to find the object-subject of sentences. And we're interested in sentences that say gene verbs a drug, or drug verbs a gene, like gene metabolizes drug, or drug activates gene. These are sentences of intense interest to those of us who care about pharmacology. One of the things we can do is we can recognize genes and drugs, and we can recognize and diseases, and we can recognize the relationships between them. So in this case, we have an ABCB1 polymorphism that influences methotrexate sensitivity. In a special little trick that I'm not going to discuss, we have also created a standardized vocabulary so we can map these arbitrary phrases into formalized phrases that are controlled terminologies. So this is a controlled term called variant. We've mapped influences to the word effects, and we've mapped methotrexate sensitivity to the concept methotrexate drug sensitivity. What that enables us to do now is go through the entirety of Pubmed and look for every single sentence that has a verb and a gene as the subject or the object, as well as the verb that relates, did I say a noun? We can look for all the sentences with a gene or a drug, and we can look for the verb that relates them. And when you do that to Pubmed, you find 170,000 different relations between genes and drugs after the normalization with the controlled vocabulary. So actually we're up over around 500,000 sentences before, but of those 500,000 of them, many of them are saying the same thing, and they boil down to 170 things that are saying different things. And so what you see here, and I should add, if it looks familiar, it's the graphic that was on the advertising poster for this lecture, produced by my student Beth Percha. We have a network that has drugs as green circles and genes as red circles. And this, if you will, shows you the complete knowledge of gene-drug interactions at the level of Pubmed. And it's complicated. There are some famous genes that we all know. CYP2D6 is a hub that affects many, many drugs. CYP3A4 is as well, and there are others. This is actually only a piece of the network. You can tell because there's only about 30 or 40 genes shown here. But each of those sentences has a drug, a gene, and a relationship. Let me zoom in on a piece of which we have the drug verapamil and the gene ABCB1. This is also known as MDR, multidrug resistant transporter, or P-glycoprotein. And here's a sentence that we found after normalization, which is that the drug metabolism of verapamil is influenced by polymorphisms in ABCB1. And that's all represented in the computer in a controlled way. Verapamil is metabolized by ABCB1. Verapamil treatment causes repression of ABCB1, and verapamil regulates the gene product ABCB1. So we've learned quite a bit about verapamil and ABCB1, and it's all now, I'm happy to report, in a computerized, computer-legible form. So now we can do some magic, or informatics. So we can chain together two gene-drug relationships to look for unrecognized drug-drug interactions. So if each of the links in my network is a drug-gene connection with a relationship, then if I have a drug that interacts with a gene and another drug that also interacts with that same drug gene, that would be two of these linked together, that might create, depending on the type of relationship that is found between those drugs and the gene, that might be an indication of a drug interaction. So Beth went to those 170,000 gene-drug interactions, provided a whole bunch of known drug-drug interactions, and learned the kinds of genes that occur in the middle and the kinds of relationships that occur on the sides in known drug-drug interactions, and then asked, are there any other ones that are unknown but match the pattern? So, for example, if this drug is metabolized by this gene, and this drug induces this gene, then you might expect that these drugs would have an interaction. That's the kind of thing she would have learned by looking at the positive examples. So when she did that, she had a long list of drug-drug interactions. She's not a physician. All the people who are not physicians, their favorite use of their advisor, me, is to walk into my office and give me these long lists and say, is there anything useful here? Now, what she did, thankfully, before she walked into my office, she went to some known online resources with drug-drug interactions. And all the ones in white here are known to be drug-drug interactions. So she did not use them in training, but she predicted them and they're already known. So we don't get any credit for a discovery because it was already known. For us, it's a confidence builder because we did not provide that information. And you can see there are many, many positive examples. The ones that are in either orange or yellow, and the color difference is not important now, it'll come up at the end, are ones where the strength is just as strong as the other ones on this list, but these have never been reported. So these are predictions. So let me just take you through one of those predictions and where it comes from. So metoprolol is a beta blocker, and dextromethorphan is a cough suppressant. It's an enantiomer of codeine, I believe, or morphine. So one of the sentences in normalized form is that metoprolol is metabolized by CYP2D6. And that comes from this very long sentence. Metoprolol and dextromethorphan were primarily 2D6 substrates, and propranolol, blah, blah, blah. I should mention that even though dextromethorphan is mentioned here, that's not what we take out of this sentence written in 2000. We just take out that metoprolol is metabolized by 2D6. In a different abstract published in 2005, there's a sentence that says that 2D6 also metabolizes dextromethorphan. So Beth has simply chained them together to say they're both metabolized by the same enzyme. There are a lot of other features that look good based on the training set. I'm going to call this a high confidence prediction. And in fact, if you look at all the genes in all of those sentences that we found, 2D6 is clearly the one that's doing most of the work in the relationship between dextromethorphan and metoprolol. So now we are going to electronic medical records in a way that I'll describe now to see if we can validate her predictions by looking for patients who are on both of these drugs and seeing if they have unexpected side effects or even expected side effects. Okay, my final story is now at the population level. Population-based and clinical data for generating hypotheses about drugs. FDA, as you may know, releases adverse event reports regularly, every quarter. But these are very complex because for each patient it gives all the drugs they're on, all the diseases they have, and all the side effects they experienced. So many people look at this and say, this is a mess, there's nothing here of value. But we thought that maybe there was some value there. And we also are intrigued by the possibility that electronic medical records do have information about patient drug exposure and the effects. So can we mine these two clinical sources to discover new drug-drug interactions? So what did we do? We first went to the FDA database and we built a statistical model to distinguish glucose-altering drugs based on an adverse event signature. So here we have a bunch of diabetes drugs and a bunch of other diseases which patients who have diabetes also have, but that are not diabetes. That's there's a reason for that. I won't go into it. The important thing is along the column, we have all the adverse events reported in the FDA database. And what we looked for were adverse events that were enriched in diabetes drugs compared to the controls. Please just think of these as controls cases. And so here you see that there's a heavier darkness compared to here, as well as here, here, and here. So these are the adverse events that we're seeing in abundance in diabetes drugs. And we use that to create a little signature. The first thing we ask is, does that signature recover all of the other diabetes drugs? And with 93% accuracy, I can tell you if something is a diabetes drug based on its side effects. And the side effects are shown here, and they might not be what you expect. Some are what you expect, like diabetes out of control, or paresthesias, depression, hypoglycemia, pyrexia, diarrhea, anorexia. Some of these are enriched in diabetics, and some of them are de-enriched or more rare in the red ones. But this is a very good signature for drugs that alter glucose, including diabetic drugs. So then my student Nick Tatanetti had a very clever idea. Why don't we take patients who are on 2 drugs, neither of which is a diabetes drug, but let's see if they have glucose-altering effects, either up or down. So he took all drug pairs, he applied the signature, and he walked into my office one day and he said, Russ, there are 4 pairs of drugs that, as far as I can tell, have nothing to do with diabetes, but where I'm seeing a glucose signal. And they were these, and the one that caught my attention was paroxetine or Paxil, the antidepressant, and pravastatin. And the reason they caught my attention is I knew that there are lots of people on these 2 drugs. And I said, well, if that's real, that's interesting. And it turns out we subsequently learned that there are about 15 million Americans on each, and probably about 500 to 800,000 Americans on both of those 2. So the first thing we did is go to the Stanford EMR, electronic medical record, and we looked for patients who were on one of these Ps, I call them P and P, paroxetine and pravastatin, who was on one P and had a glucose measurement, and then got the other P and also had a second glucose measurement, all within 60 days. So that's pretty strict, and we only found 12 patients. But at Stanford Hospital, those 12 patients had a pretty impressive increase in their glucose. Now, but it's 12 patients. So yes, I'm an informatics guy, but I'm not an idiot. I know that that's an intriguing initial result, but is not probative of anything. And in fact, we looked at Paxil alone and pravastatin alone, and they were pretty flat. And the combination did seem to go up, but there was some funky differences in the baseline, and 12 was just not enough. Look at those error bars. So I called up my friends at Vanderbilt and at Harvard, showed them these results, and said, hey, could you guys go into your electronic medical record? This is Dan Roden and Zach Kohani. Could you guys go into your electronic medical records and check? And so Vanderbilt came back. They had 20 patients, and they saw the same effect. And then Harvard had 106, and they really saw the effect. And so I think altogether, I don't have the altogether side, but altogether, it's a very clear, tight error bars, 20 milligram per deciliter increase in glucose in this retrospective analysis of the electronic medical record, with the individual drugs alone over 60 days showing no bump. We also looked at other combinations of antidepressants and cholesterol drugs, and none of them had any significant bump. This one had an insignificant bump, barely significant, barely insignificant. But really, this is very specific to those 2 drugs, which we don't understand yet. And there, really most concerning was the fact that in diabetic patients, the rise in glucose was 50 milligrams per deciliter, also with no change in baseline when you start one drug or the other drug alone. So we submitted this paper. It was a little rocky because it was entirely computational, right? We did this crazy data mining thing to get the hypothesis. Then we did data mining to kind of get evidence in favor of it. I'm happy to report that the paper was accepted just based on this data, but we knew that people would want to see in vivo data, so not knowing what would happen to the paper, we exposed mice to the drugs as well. So what you see here are mice on just pravastatin, mice on paroxetine, 2 different diet controls, high fat and regular, and then mice who were getting both paroxetine and pravastatin. And so we added this to this paper after as a note added in proof, but this does seem to be true in Vanderbilt, Stanford, and Harvard patients and mice. And so we're following up because we don't have a mechanism. So really I've said what I wanted to say at a high level. I think that the informatics, we're excited because they really do provide these powerful tools that allow us to do discovery at the molecular, cellular, and organism level. I've told you separate stories there. I think it's exciting that they operate on primary data, but they can also operate on process knowledge such as text. They can individually contribute to our understanding of drug action, but I think the combination is where we all need to move. And so let me just say something about that. The orange guys here, I didn't tell you what they were before. These are things that Beth predicted from her text mining, but that Nick, the guy who did the mouse studies and the paroxetine, also saw in the FDA data. So if I were a betting person, and I definitely am a betting person, if you ever meet an informatician who said that they're not a betting person, they're lying, then I would say that these are much more convincing than the 2 yellow guys, because I now have 2 entirely different lines of evidence. The biological text and the FDA data mine and subsequent EMR validation. So we're looking very carefully at naproxen and Valium, verapamil and omeprazole, and verapamil and fexofenadine, but we have 2 different modalities. Similarly, we are looking at the drug repurposing, the molecular data, and we're looking at that expression data to look for pathways that are affected. So here we have a known drug-target interaction. We've predicted interactions based on similarities in the pocket of the protein, but now we're percolating that down to the expression data to see what diseases might be affected, or if the affected diseases show sensitivity to these new drugs. So I think this is just to show you that it has not been lost upon us that whatever you can do with the individual data sets, the combination is really where the future is. And so I want to leave you with the promised emerging network. So here we have the 3D structure, we have drug recognition and binding. All can be done with publicly available databases. We have the cellular responses available from Geo. We have the text mining. We have the population database mining from the FDA, and we have the electronic medical records. And now for each one of these lines, it means a conversation between students in my lab or to themselves, thinking about integration experiments that bring this together. But what results, I think, and I hope I can convince you of this with what I've said, is a really powerful network of data that allows us to do a lot of work in the computer before we ever touch a mouse. We did those mouse experiments, and how confident was I? Was very confident that we were going to see an effect. Was I sure? Of course not. But the point is, those were focused experiments. That entire mouse experiment cost me $3,000, and I only had to spend that money when I had a very focused hypothesis. And so I think that the real message here is you can do not only hypothesis generation, but initial triage, so that your experimental work can be extremely focused and therefore cost-effective, and therefore hopefully catalyze and accelerate science and the efficiency of science. So with that, I want to stop. I want to thank you. Here's the various websites. One of the things that Symbios does is put out a quarterly magazine called Biomedical Computation Review, where we try to celebrate the joys and the miracles of biomedical computation. It's online, but you can also get a hard copy, and you can go to the website and request that. So thank you very much for your attention, and I think we have some time for questions. Yes.
Hi, that was a great lecture. And so I'm very curious about the text mining. Did you put weights on connections that you saw multiple times in 5 or 6 journals? And did you exclude weights that you saw just once or twice, even though, it might be new, but it still might be real?
Yeah, so that's a great question. So what I thought you were going to ask, so I'll answer the question you didn't ask, which is, do you weight New England Journal higher than a solid disciplinary journal? And the answer is no, although we've thought about that. But I'm not sure which way the weights would go. That's a different discussion. If you remember, we actually, for any gene-drug interaction, we often have hundreds of sentences that relate them, and so we don't really have to weight them because the number of times it's said feeds into the machine learning algorithm, and it notices that certain words and certain relationships occur more often. So one thing we're worried about is whether we should actually remove the redundant ones, and then we would have to worry about weighting. But right now, there's kind of an implicit weighting. I want to stress that I'm sure those of you who are good scientists are saying, well, wait a minute, everything in the literature is not true, and I am totally aware of that. So that's why these are hypotheses based on things you could read in the literature, and after that, caveat emptor, and all your normal scientific skepticism has to be applied, such as asking if it occurs in two different independent analyses. So right now, no weighting except implicitly by the frequency of the sentence.
And real quick follow-up, do you think going into the actual journal instead of just looking at the abstract would give you more or less information, or do you think it doesn't really matter?
So there's a literature on this, and it turns out that you can get misled by the sentences in the discussion because people speculate more, and that might lead to more false positives. But you definitely also get more true positives because there's more time to mention more true things. So we would love, and with the growth of PubMed Central, this experiment will be doable pretty soon. So we're big fans of the PubMed Central because it gives us full text access, but we haven't taken advantage of it yet.
Okay. Thank you very much.
Thanks. I guess we'll just bounce back and forth.
So the ICA approach to expression data is impressive. I'm wondering if it's been applied to GWAS data. That's probably an order of magnitude more available public data, and we're missing a lot of the genetic components of many diseases.
Yeah, I think that's a great idea. We haven't done that because the number of GWAS for drugs is just beginning to come up, and I try to keep the lab focused, right? Because there's a lot to do, and as you can see, I don't know if you call that focused, but at least every project has the word drug in it. So I'm aware of a number of GWAS for drugs that are coming out, and then it might be relevant, but we haven't done it yet. Yes?
So what percentage of the drugs you think you might have covered, because we still don't know so much about other genes and their protein function? You mentioned a few of them.
So I think that these cellular based assays are incredibly powerful for a data-based, unbiased view at drug response. The problem, of course, is that many drugs might not act via transcriptional control. They might act entirely at the post-transcriptional level, so that immediately makes you a fan of proteomics and metabolomics, in theory. The problem, as I think we all know, is that those are still promising emerging fields, but they haven't crossed that threshold where we can get large amounts of reliable digital data. When they do, I can guarantee you that my lab will be looking at those data sets trying to pull things out, but I think that's a huge shortcoming of only looking at the expression data. Also, the GWAS and genomic data I think is also very powerful, so we shouldn't be complaining. We do have a richness of information, but for drug response, I still think you're right that we're still challenged with respect to the full amount of data that we would like to have.
And also there are drugs which are really pro-drugs, like
Yes.
Plavix.
Right.
So where do you stand there? Basically, you'll be looking at the metabolites rather than the drug itself.
Right, so that's where metabolomics comes in because you can measure those. There are some interesting progress in creating computational livers. These are, say, models that will take a drug and will apply the various transformations that the cytochromes are able to do, add an oxygen here, an oxygen there, to come up with credible hypotheses about what the metabolites may be. And so I think another area, and I haven't really stressed it today, but this area of chemoinformatics, which our colleagues in chemistry have been thinking about computational methods for handling their huge libraries. And I think it's very exciting to think about bringing biology and bioinformatics together with chemoinformatics. We don't have any results in my lab yet, but we have some projects now where we're starting to look at molecules not as a name like verapamil, but as a 2D or 3D structure, and thinking about what the metabolites might be and which pockets they might be able to bind.
But then there are also blood-brain barrier effects. So whatever happens in systemic level, could it interact also with the...
Right. So you're absolutely right. I mean, so is drug response complicated? Yes. So, and I don't think we're going to have everything solved anytime soon. Yes.
I think your work integrating the multiple data sources to form hypotheses is really excellent. I work in drug discovery and functional genomics, and one of the big frustrations is that many screens are being done now, and they end up as supplements, either a PDF file or an Excel file on the back of a publication. Going forward, how do you see the field kind of bringing that data to the mainstream, like what's been done with the expression data sets?
Yeah, that's a great question. We have two examples where things had three examples. So three-dimensional structural data goes to the PDB, nobody asked questions. I've talked to GMS veterans who said that that was not a trivial thing to achieve. Gene expression data, for the most part, has been going into GEO, although there are logistical issues now with RNA-seq that the size of these databases, be careful what you ask for. And I think genetic and GWAS data with dbGaP is pretty good, and of course PubMed and others. But you're right that as these fields are emerging, especially if it's a screen that is not of a standard type, and as you know, a lot of the drug screens are very peculiar for the application area of interest. It's hard to imagine how to create a database of such things when they might be a one-off experiment. Even though if it's 5,000 or 50,000 measurements, it's the only time that the data is going to be in this exact format. And so my informatics friends and I think about this a lot because it is definitely technically not difficult to come up with a language that describes the data, and then you produce the data through XML and things like this. But you need to have the political will and scientific will to take that extra step to make the data available. And so many scientists, the moment the paper is accepted, it's like, thank God that's out, let's move to the next thing. And so that kind of very unsexy, unsatisfying activity of formatting data for future generations just gets lost. I think there might be a role for journal editors and funding agencies.
Okay, thank you.
Without using too heavy of a hammer. Yes?
So at the beginning of your talk, you show us the database of drug-resistant SNPs, and then you followed with the future balloons slide. And I was kind of almost waiting that the next slide will come, how those SNPs affect those future balloons, and it didn't. So
we're very interested in this. So the question is a good one. If we're going to have information about human variation that affects drug response, and if we're going to have 3D structures, why don't we put them together and have studies about how the 3D structure changes in the context of an amino acid change, and how that might explain the phenotypes. And I'm happy to tell you that one of the postdocs in my lab is doing that specific project on the warfarin target. So VKORC1, there was recently a bacterial homologue published from bacteria, obviously. She has built a human model of the bacterial homologue and is now starting to mutate the sites for the known human variations and seeing if she can predict the phenotype. So I absolutely agree with you, just didn't talk about it today. Good, perfect timing.
Let's thank Dr. Altman again. And please join us in the library for refreshments. And before you go, please note that the next Walls talk will be on November 2nd by Dr. Kenneth Fishbeck. Thanks all very much.